Welcome back, and now that you know some of the basics of genetics, we're going to discuss what specifically a monohybrid cross is. Now, a mono, if you know from our prefixes, is one. So a monohybrid cross is actually where we're going to take genes from one parent and another parent, one set of genes, okay, one gene from one parent and one gene from another parent. We're going to cross them and we're going to determine what that means for their offspring, okay? So we're going to talk about the term segregation. What does that mean, first of all? And then, what do the terms genotype and phenotype mean? And then finally, we're going to talk about the steps in a monohybrid cross and how is it that we can complete the Punnett square to figure out what the genotype and phenotypes of the babies might be. So our first piece is segregation. Segregation is just separating something. Okay, you may have talked about segregation in your history class, okay? But segregation is just a term that deals with separating of something. So if we're going to separate this, we're actually talking about the separation of genes. So each person actually carries two alleles for a specific gene. You get one set from mom and one set from your father, and you now have two sets of those genes. But to be able to pass those on to your offspring, you can only pass on one set of those. So meiosis, which we've talked about in the past, is the process that actually takes those chromosomes, those two chromosomes that have the same genes on them, and separate them so that parents are only passing on one allele to each offspring. Now, because of that, if we have parent number one here, let's say parent number one has a heterozygous genotype, so big G, little g. It could pass on either the big G or the little g, but it's not going to pass on both. So if this is our, this is parent number one, these would be the reproductive cells of that parent number one for that single gene. Parent number two, if parent number two is also heterozygous, that parent number two could pass on one of the two, either big G or little g. A Punnett square, which we're going to look at in another 2 slides or so, is going to show us the difference between what will happen if parent number one passes on the big G and parent number one passes on the little g, or if parent number one passes on the big G, parent number two passes on the little g, or any other combination that could happen here between parent number one and parent number two. Okay? So the big key terms for today are genotype and phenotype. So what do those letters mean? What do the letters mean if we have big G, little g, or whatever it is? So the genotype, the letters actually stand for the genotype. Okay? The genotype are the two alleles that you carry, and we use those letters to represent it. So you could be homozygous dominant, big G, heterozygous, big G, little g, or homozygous recessive, little g. Okay? So the genotype are the actual genes that you carry, whereas the phenotype is the actual physical traits that you're going to see on that organism. So does that organism have brown hair? Does that organism have blonde hair? Do they have attached or unattached earlobes? And there are some genotypes that would be different genotypes, but would show the same phenotype. Okay, so that's why we distinguish between genotypes and phenotypes. So let's take a look at this here. So if we're looking at earlobe shape, unattached earlobes are dominant and attached earlobes are recessive. So we've used big E for unattached dominant earlobes and little e for attached recessive earlobes. So we have 3 different genotypes we could get. We could have capital E, we could have capital E, lowercase e, or we could have lowercase e, lowercase e. Now, if remember what we said before, if you show the dominant allele, what's going to happen is that you're going to actually show that dominant trait on the physical person. So both capital E and capital E, lowercase e are going to show unattached earlobes on somebody's ear. The only way to get attached earlobes is if you have lowercase e as your genotype. Okay, so genotype and phenotype. Genotype, what are the genes that you have? Phenotype, what does it actually look like on your body or on the organism? So in terms of that, we're going to take a look at, okay, so how do we pass those traits on to offspring, and what does that actually mean? So a Punnett square is going to show us the probability of two parents passing on traits and what the probability of those genotypes and phenotypes are with those offspring. Okay? So first thing you need to do when trying to figure out a problem is to fully read the question. You need to determine the trait that's being shown, and you need to figure out what are the parents' genotypes. So the first thing here, and I'm going to do some highlighting or underlining, I should say, on here. It says a heterozygous green pea plant is crossed with a yellow pea plant. Green pods, big G, are dominant to yellow pods, little g. So we have two parents here. We're crossing a heterozygous green pea plant, and we're crossing that with a yellow pea plant. Well, the problem tells us that green pods are dominant to yellow pods, big G and little g is what we're going to use. I will also give you a hint here. If it doesn't give you the letters, if it doesn't tell you what's dominant and what's recessive, if something tells you that it is heterozygous green, remember, that has to be the dominant trait. Okay? Heterozygous always shows the dominant trait if we're talking dominant versus recessive. Okay? So, parent number one is heterozygous green, which means it has to have a green, so a big G allele, but it's heterozygous. Because it's heterozygous, it has two different alleles, so it must have a big G and it must also have a little g or a yellow allele. Parent number two is yellow. Now it doesn't say whether or not it's homozygous or heterozygous, but your key here is that yellow is recessive, so the only way to show yellow is if it has two yellow alleles. So parent number two must be little g, little g. So now that we've figured out the parents from the problem, the second step is to take those parents and set them up in a Punnett square. So parent number one that we were just looking at was big G, little g, and parent number two was little g, little g. Now notice, as I put these parent genotypes in the Punnett square, for this box we have parent number one that I put across the top and parent number two that I put down the side. You're going to always put one parent across the top and one parent down the side. Do not mix them up. If you do that, your crosses are not going to come out correctly. The second thing, after doing that, you put one gene across the on the top of one box and one gene on the top of the second side of the box. And then you're going to fill in the Punnett square. To do that, you're going to take, each box is going to now be filled with two letters. Okay? One it got from its mother and one it got from its father. So to do that, you're going to look at the top, the box that's the letter that's on the top of that box, and the letter that's to the left of that box. So at this box, we have a big G at the top, and we have a little g to the left. Whenever writing out a genotype, always write capital letter before lowercase letter. It just helps you see it a little bit easier as to if it has a dominant trait or not. Okay? So this box right here would carry big G, little g. This box would have little g, little g. This box would have big G, little g. And this box would have little g, little g. Once we've filled out the boxes, we're then going to take a look at our Punnett square and determine what genotypic and phenotypic ratios we would get for our offspring. So on the next slide, I've re-put the Punnett square up here. Okay? And so we're going to figure out genotypic and phenotypic ratios. So remember, we just said genotype are what are the genes that it carries, what are the 2 letters combinations that we have. So the first thing we need to do is look in the box and figure out what are the different letter combinations that we have. We have big G, little g. And we have little g, little g. So we've only got those 2 combinations. So we're going to write that down. And then the next thing we need to do to figure out the ratios of this is how many big G, little g's do we have and how many little g's do we have. So in counting them out, we have 2 big G, little g's. And we have 2 little g's. So we can write it out like this, or we can reduce it and it means the same thing. So you can get 1 big G, little g to every 1 little g. So what this means is that for our offspring, we should see about 50-50%. 50% big G, little g, and 50% little g. Probability is not an absolute, okay? If I flip a coin, I have a 50% chance of hitting heads and a 50% chance of hitting tails. If the first time I flip my coin, I land on heads, the second time I flip it, I still have a 50% chance of hitting heads and a 50% chance of hitting tails. So I could hit heads 2 times in a row and have 100% and 0%. The larger the set of numbers you're looking at, the more closely you're going to get to that 50-50 ratio or whatever the ratio is that you have. So the second one is to determine the phenotypic ratios. So we said big G is green, little g is yellow. So big G, little g would show the green phenotype. So 1 plant that produces green pea pods for every 1 plant that produces yellow pea pods. Remember, little g is recessive, so little g would be the recessive trait and would show the recessive. So 1 for every 1 yellow genotype, yellow pea pods that are produced. So this is how you figure out a Punnett square. Let's take a look at a second problem or workout on this. So our first step again, figure out what the parents are. A heterozygous green pea plant is crossed with another heterozygous green pea plant. What are the genotypic and phenotypic ratios of the offspring? So we're trying to figure out genotypic and phenotypic ratios of the offspring. Parent number 1 is heterozygous green pea plant. So heterozygous green, we already said, would be big G, little g. And this is crossed with another heterozygous green pea plant. So parent number 2 is also going to be big G, little g. Okay? So we take parent number 1 and put them across the top. Big G, little g. We'll take parent number 2, put them down the side. Big G, little g. And then when we fill in our Punnett square in the center, you'll notice that our probabilities and ratios are going to be different than what we just saw because our parents are different than what we saw. So this box will be big G, little g. This box, big G from the top and little g from the side. And then this box will be 2 little g's. So now as we look at our genotypic and phenotypic ratios, you're going to notice that we have a big G. We also have a big G, little g. And we also have a little g. Okay? We have 1 big G. We have 2 big G, little g's. And we have 1 little g. So our ratio for this is 1 to 2 to 1. Okay? 1 big G for every 2 big G, little g's for every 1 little g. Now our phenotype ratios are going to be slightly different then because we're going to have 2 combinations here that could produce green pods. Both our big G, big G, and our big G little g can produce green pods. So our phenotype ratio is going to be 3 green to every 1 yellow. Okay? So this is how you fill out a Punnett square and complete a monohybrid cross to determine what the genotypic and phenotypic ratios of the offspring of two different parents are going to be.